What is genetic engineering? Genetic engineering is the manipulation of an organism's genome through the use of biotechnology. One example of genetic engineering is using the GFP gene as a reporter of expression. GFP is taken from the jellyfish Aquaria victoria, and it absorbs blue light and emits green light. In other words, it has green fluorescence. GFP is introduced to organisms to tag certain genes or proteins. GFP is introduced to bacteria, such as E. coli, via transformation. Transformation is the process in which bacterium take up DNA from the surrounding environment and incorporate it into their own DNA. Transformation results in genetic information that is both stable and heritable.
Our hypothesis was that the plate that does not contain DNA but has LB medium will have growth but no glow. The plate that does not contain DNA but has LB and ampicillin for the medium will have no growth and no glow. The plate that contains DNA but has LB and ampicillin for medium will have some growth and will not have a glow. The plate that contains DNA and has LB, ampicillin, and IPTG will grow and glow due to IPTG.
500 microliter of ice-cold calcium chloride solution is added to the negative test tube. Using a toothpick, about 15-year-old isolated colonies from the E. coli source plate is transferred into the negative DNA test tube.
Yeah, go ahead. I don't know if we have a lighting system. No. There's different lights that you can use. So I didn't do a good job of taking the single. These are really shoddy dogs.
The toothpick is being twisted to freeze the cells into the tube.
Make it focus, yo. There we go. Yeah, well, that one got a lot of colonies. This one got some colonies. This one's got a lot on there too, but people have been stabbing this one. So. Oh, okay. That's a good one. This one, you don't have to be as careful because after.
Mixing the solution. Incubating both tubes on ice for 10 minutes. The cells are being heat shocked for 90 seconds in a 42 degrees Celsius water bath. 250 microliter of each solution is being added to each plate. The cells are being spread over the entire plate with a sterile inoculating loop.
I'm so frightened of ripping it apart.
You know, record. Just be gentle.
The plates are covered and the liquid is allowed to be absorbed for 5 minutes. The plates are placed in an incubator at 37 degrees Celsius for overnight incubation. All plates are placed in an inverted position.
Our results showed the following. For the plate containing PFG, plasmid DNA, ampicillin, and IPTG, our results showed growth of bacteria that glowed under UV light. For the plate containing PFG, plasmid DNA, and ampicillin, our results showed growth, but there was no glowing. For the plate without the PFG, plasmid DNA, but with ampicillin, there was no growth. And for the plate without DNA or ampicillin and containing only LB, our results showed significant bacterial growth. The PFG, plasmid DNA, ampicillin, and IPTG plate had growth because the gene gave the bacteria ampicillin resistance and the IPTG inactivated the repressor on the gene. IPTG is an allolactose mimic which allows the bacteria to express the PFG gene. The plate with DNA and ampicillin had growth because of the ampicillin resistance in the gene, but it did not glow because there was no IPTG that allowed for the expression of the PFG gene. For the control cells, the plate without DNA but with ampicillin showed no growth because these bacteria did not receive the ampicillin resistance in the gene. The plate without DNA or ampicillin showed growth because there was no ampicillin that could kill them, and they had LB, which is a broth that allows bacteria to grow easily. The only plates with genetically transformed cells were the plates with PFG DNA that were not the control cells. The purpose of the control cells was to show that E. coli could grow under the conditions given to them, and to show that ampicillin could be an effective way of selecting for only the bacteria that picked up the gene we wanted them to pick up. Our calculations for transformation efficiency showed that for every microgram, 200 transformations took place. These results supported our initial hypotheses for finding which bacteria plates would grow and which ones would glow. The end.
La fin.
Payan.
Fin.